Hello young guys, good evening. You are welcome in the SOAP classes. Today we will talk again about carbohydrate metabolism part 3 and tomorrow we will talk about part 4. It is for students of higher classes. So we have discussed already the glycolysis and Krebs cycle. One more pathway is there in some animal cells and in some plants where it is called HMP shunt or hexose monophosphate shunt. It has four importance. First of all, it generates NADPH instead of NADH of normal glycolysis. Here NADPH is produced and it is used for synthesis of fatty acids and steroids from acetyl coenzyme A. Number one. Number two, it is also important for the synthesis of 5 carbon sugars which can be utilized for formation of nucleotides and nucleic acids. Third, it is also helpful for complete oxidation of 5 carbon sugars through 5 carbon sugars are converted into 6 carbon sugars. In the step 2, the 6 carbon sugars are converted to 5 carbon sugars. Here maybe just 6 carbon sugars may be formed from 5 carbon sugars and then they can undergo glycolysis. So this way 5 carbon sugars can also be oxidized by glycolysis and Krebs cycle. And the fourth stage is that the glucuronic acid formed in this and some 5 carbon sugars are intermediate steps in the dark reaction of photosynthesis. So there are two categories of reactions. First of all, what happens? It starts from glucose 6-phosphate and previously there is removal of 2 hydrogen ions, and they are taken by NADP to form NADPH. So in the intermediate step, here 6-phosphogluconolactone is formed. If you have any, you can emit this step, you can directly convert it into, but actually the step is that initially there is removal of 2 hydrogen from glucose 6-phosphate from the carbon number 1, and then again there is addition of 1 water molecule. So oxygen is added here at this point and this way it forms 6-phosphogluconic acid. So this aldehyde group is oxidized to form gluconic acid. And after that what happens? Both reactions take place at the step. There is removal of 1 more hydrogen from this and also there is removal of CO2 from carbon number 1. So when CO2 will be removed, this hydrogen remains as such and binds with this carbon number 2. It forms CH2OH, and here from the 2 hydrogens are removed, it forms the CO group. So this way a ketonic 5 carbon carbohydrate is formed called ribulose 5-phosphate. So this is one category of reaction by which first of all there is oxidation, at 2 level there is dehydrogenation, and in the last there is removal of carbon dioxide so that the 6 carbon carbohydrate is converted into 5 carbon compound, but initially it is ribulose 5-phosphate. After that what happens? That in the second step simply there is interconversion in between and by isomerase enzyme. So what happens? This was the gyrulose 5-phosphate At this level, carbon number 3, you can see here, hydrogen is located on this side, OH on this side. So there is isomerization and H comes in this side, OH on this side. It forms the ribose 5-phosphate. Then, so ribulose, xylulose, and then ribose phosphate. After that, 2 steps are there. One, there is interconversion in between xylulose 5-phosphate and ribose phosphate, both 5 carbon compounds, and there is formation of 3 carbon 3-phosphoglyceraldehyde and a 7 carbon heptulose. Sometimes one more interconversion takes place in between these 2 molecules, and a 6 carbon fructose 6-phosphate and a 4 carbon erythrose 4-phosphate is formed, which can go again into further glycolysis and Krebs cycle type of reactions. About 60% of carbohydrate oxidation, glucose oxidation in liver cells is by this mechanism called HMP shunt. Thank you very much.